Hello students, in 8th standard you learned about the basic unit of life, the cell. As a continuation, let's first talk about the discovery of cells. In the year 1665, Robert Hooke was observing a thin slice of cork under a primitive microscope. Cork is a substance in the outer layer of the bark of a tree. He observed some honeycomb-like structures under the microscope. He called them cells. But what he observed was dead cells. After him, in the year 1674, Anton van Leeuwenhoek discovered living cells from pond water with the help of an improved microscope. Let's try to understand how the cells from an onion appear. Take a small piece from an onion bulb. With the help of forceps, we can peel the layer from the concave side of an onion. This is the concave side of an onion, and with the help of a forceps, we can peel the layer from there. This layer is called the epidermis. Epidermis is a layer which gives protection to the inner body. Dip the epidermis immediately into water kept inside a watch glass. Otherwise, it will get dry and get folded. Now take a glass slide and put a drop of water onto it. Take a small piece of peel, place it on the glass slide so that it is perfectly flat on the slide. It should not get folded. Put a small drop of safranin onto this peel. Safranin is a biological stain which is red in color, and this will give red color to the peel so that we can easily observe the parts under the microscope. Now place a cover slip on top of it. When we place cover slip, we need to ensure absence of air bubbles. Now if we observe the slide under microscope, we can see the onion cells. They have nucleus inside it, and each cell is separated from one another. By this layer, these cells are separated from one another, and this is called the cell wall, and inside it you can see a small dot. This is the nucleus. If we place different sizes of onion peels, we can observe that the size and shape of the cells are same. Regardless of the size of the onion bulb, cells looks similar in their shape and size. These small cells together form the big onion bulb. In other words, we can say cells are the building blocks of onion bulb. All the organisms, whether they are small or big, are made up of cells. Depending upon the number of cells present in an organism can be divided into two: single cellular organisms and multicellular organisms. There are some organisms which have only a single cell in them. This one cell performs all the functions in the body. These kinds of organisms with only a single cell is called single cellular organisms. Example of single cellular organisms are amoeba, chlamydomonas, paramecium and bacteria. You can see a paramecium here. There are organisms with more than one cell. A group of similar kinds of cells perform a specific function, and they together form different body parts. These kinds of organisms are multicellular organisms. Fungi, plants and animals are examples of multicellular organisms. In most of the multicellular organisms, the cells that perform different functions are different in their appearance and shape. Example, smooth muscle cells that line the digestive organs are spindle shaped. Red blood cells are spherical in shape. Nerve cells are branched. There is ovum, which is a female reproductive cell, and sperm, which is a male reproductive cell. All are different in their shape and size. From this we can understand that the shape and size of the cells is related to the specific function they perform. When we have to perform a tedious task, what is that we follow? We divide the work and give a part of the work to a particular person or a group of persons. This is called division of labor. Multicellular organisms also follow this division of labor. For example, the heart will pump blood, stomach will digest the food. This division of labor is there in a single cell as well. This is an animal cell and the cell contains a number of cell organelles. All these are cell organelles present in a single cell. Each kind of cell organelle performs specific function, like some will make proteins, some will destroy the waste materials, etc. All the cell organelles together constitute the basic unit of living beings, the cell. All the cells, no matter what organisms they are in or what function they perform, they have the same cell organelles in them. If we observe a cell under a microscope, we can see three features which are similar in all cells. They are plasma membrane, nucleus and cytoplasm. Let's learn about them. Plasma membrane is a membrane that separates the inner organelles of a cell from the outside environment. Here you can see the plasma membrane. This membrane, this outside covering is the plasma membrane of a cell. Plasma membrane is made up of lipids and proteins. Here you can see an enlarged portion of the plasma membrane. We can observe the structure of plasma membrane by means of an electron microscope. Electron microscope will give high resolution images. For the functioning of cells, it is necessary that materials should enter and exit the cell, and these materials enter and exit the cell through this plasma membrane. This plasma membrane is semi-permeable. That means it will allow some materials to enter and it will block the entry of others. Let's see how the materials enter and exit a cell through this plasma membrane. Some substances enter and exit the plasma membrane through simple diffusion. Do you know what is the meaning of diffusion? It is the movement of particles from higher concentration to lower concentration. The movement of oxygen and carbon dioxide happens by means of diffusion. Inside of this plasma membrane is the inside of the cell, and this is outside area. If the concentration of carbon dioxide is high inside the cell, if concentration of carbon dioxide is high inside the cell, it must be eliminated as it is a waste product of cellular respiration. When the concentration of carbon dioxide becomes high inside the cell, and if the concentration of carbon dioxide is low outside the cell, then carbon dioxide will move from a region of higher concentration to a region of low concentration. That means as soon as there is a difference in concentration, carbon dioxide move out of the cell. Same is the case with oxygen. Oxygen is essential to carry out the functions inside a cell. If the concentration of oxygen is low inside the cell, and if the concentration of oxygen is high outside the cell, then the oxygen will move from a region of higher concentration to low concentration through this plasma membrane. That means oxygen will enter into the cell. The exchange of carbon dioxide and oxygen is called gaseous exchange, and this gaseous exchange is happening by a process called diffusion through plasma membrane. The movement of water is also through diffusion. The movement of water molecules through a selectively permeable membrane is called osmosis. The movement is affected by the presence of substances dissolved in water. Let's try to define osmosis. Osmosis is a passage of water from high concentrated region. If water concentration is high outside and water concentration is low inside, then water will move into this cell through the plasma membrane. Osmosis is a passage of water from a region of high concentration to a region of low concentration through a selectively permeable membrane till equilibrium is reached. Equilibrium means both the regions reach the same concentration level. If a cell is placed in a solution which contains exactly the same concentration of water as inside the cell, there will not be any change to the cell. This cell is placed in a solution. If the concentration of water in the solution and concentration of water inside the cell is equal, then there will not be any change to the cell. The amount of water transported into the cell is equal to the amount of water transported from the cell. As a result, the cell will stay in the same size. Such kind of solution is called isotonic solution. In isotonic solution, the concentration of water is same as that of the concentration of water inside a cell. If we place an animal cell or plant cell into a solution containing sugar or salt, let's see what will happen. Let's make a diluted sugar solution by adding a small amount of sugar to water. In this diluted solution, the concentration of sugar is less and concentration of water is high. Now let's place a cell inside this diluted sugar solution. Diluted sugar solution, the concentration of water is high compared to the concentration of water inside the cell. The concentration of water inside the cell is less. As a result, the water molecules will enter into the cell from a high concentrated region to a low concentrated region. As a result, the cell swells up. The cell gains water as a result of osmosis, and such kind of solution is called hypotonic solution, where the concentration of water is higher than the cell. Now let's make a concentrated sugar solution by adding more sugar into water. Here in the concentrated solution, the concentration of sugar is high and concentration of water is less. Now let's place a cell into this solution and let's see what will happen. This cell is placed in a concentrated sugar solution where the concentration of water is low, and inside the cell the concentration of water is high. Then water will move from a high concentrated region to a low concentrated region. The water will move from the cell to the outside solution. As a result, the cell will shrink. Such kind of solution is called hypertonic solution. In isotonic solution, there will not be any change to the cell. In hypotonic solution, the water will enter into the cell and the cell swells up, and in hypertonic solution, water oozes out from the cell and the cell will shrink. From this, we can conclude that the osmosis is a kind of diffusion through a selectively permeable membrane. Let's do some more experiments. Egg is a single cell. Egg has an outer hard cover and an inner semi-permeable membrane. To see osmosis, we have to remove the outer hard cover of egg. For that, let's place egg in vinegar. Take 2 eggs and place them in vinegar. The vinegar contains acetic acid. The outer cover of egg contains calcium carbonate. When both of them come in contact with one another, then the calcium carbonate and acetic acid reacts with each other. As a result, carbon dioxide, water and calcium acetate is formed. We could see bubbles inside the solution due to the production of carbon dioxide. As a result of this chemical reaction, the outer hard cover will get dissolved. Now we got two eggs with a semi-permeable membrane which protects the inside of this egg. Now place an egg in a hypotonic solution, that is water. Water is a hypotonic solution as it contains a smaller number of dissolved solutes and the concentration of water molecules is high. The water concentration is high compared to inside of the cell. Here the concentration is high and here the concentration is low. Then water will move from a region of higher concentration to a region of lower concentration. That means water will move into the egg and egg will get swelled. Then place another egg in hypertonic solution. We can prepare a hypertonic solution by adding more salt into water. Then the solute concentration is high and water concentration is low. The water concentration is low outside and water concentration is high inside the egg. When we put the egg in this hypertonic solution, water will move from a high concentrated region to a low concentrated region. That is, water will flow from the cell to outside solution. As a result, the egg will shrink. We can observe this osmosis in our daily life. Have you observed that if we place raisins in water, it will get swelled? This is due to osmosis. The water enters from a high concentrated region to a low concentrated region through the plasma membrane. As a result, the raisins get swelled. Cells get nutrients from environment by means of diffusion. Plant roots absorb water by osmosis. A few minutes before, we learned about the structure of plasma membrane. The flexibility of plasma membrane enables the cell to engulf the food from outside. Do you know how amoeba eats? Amoeba acquires food through a kind of process called endocytosis. In endocytosis, the material to be engulfed is surrounded by an area of plasma membrane. Here you can see these are the food particles and this is the plasma membrane of amoeba. These food particles are engulfed by a portion of this plasma membrane. And after this area of the membrane buds off inside the cell as vesicles. You can see here the vesicles and inside the vesicles the food particles are there. These vesicles are transported deep into the cell. This process is called endocytosis. Then these vesicles come in contact with a cell organelle called lysosome. This is lysosome and lysosome has digestive enzymes inside it. These digestive enzymes digest the food inside the vesicles. This is how amoeba eats with the help of endocytosis. Thus, we saw plasma membrane is a semi-permeable membrane that allows some materials to pass through and block others. Now let's learn about the cell wall. In plant cells, in addition to plasma membrane, there is another rigid outer covering. It is called the cell wall. Here you can see this is the plasma membrane. This layer is the plasma membrane and this is the cell wall. This is the rigid cell wall outside the plasma membrane. It provides the cell with structural support and protection. The cell wall also acts as a filtering mechanism and is made up of cellulose. Now let's learn plasmolysis. Let's place a cell in hypertonic solution. In hypertonic solution, the water concentration is low. The water concentration is low outside the cell and high inside the cell. As a result, the water will flow from the cell to the outside solution. This will lead to the shrinkage of the cell. Here you can see the cell is shrinked. This process is called plasmolysis. Plasmolysis happens when we place a cell in a hypertonic solution. Then the water will move outside from within the cell, and as a result the cell shrinks. Let's do one activity to understand more about plasmolysis. Take two Rhoeo leaves. Take a small piece from inside the leaf and place it on a glass slide. Take another piece and keep it on another glass slide. To the first leaf add few drops of water, and to the second leaf add few drops of salt solution. Place cover slips and observe under a microscope. In the first slide, we can see the water is getting into the cell and the cell is swollen as it is placed in a hypotonic solution, that is water. Water gets into the cell and the cell is swollen. But in the second slide, as the cell is placed in a salt solution, which is a hypertonic solution, water from the cell oozes out to the surrounding medium and the cell shrinks. We can see plasmolysis here. The water oozes out of the cell and the cell shrinks. This is a plasmolysis. The cell is able to absorb water as it is alive. What if we place a dead cell in a hypertonic solution? Let's observe. Now take another leaf and place it in boiling water. The cells will die. Now add salt solution to it. Observe under microscope. We cannot observe any change as the cell is dead. From this, we conclude that dead cells don't perform plasmolysis. The cell wall can protect the cell if they are placed on a dilute solution, that is a hypotonic solution, from bursting. When the cell is placed in a hypotonic solution, water will enter the cell and there is a chance that the cell might burst. The cells swell and it builds up a pressure against the cell wall, but the cell wall exerts an equal pressure against the swollen cell. Thus, the cell wall can save the cell from bursting due to osmosis, but this protection is not there for animal cells as the cell wall is absent in animal cells. Now let's learn about another important cell organelle, the nucleus. Earlier I spoke about an experiment where we took onion peel and colored it with safranin and observed it under microscope. We can use iodine or methylene blue as dyes. According to the chemical composition, different regions of a cell get colored differently. Here you can see inside the cell some areas are very lightly colored and some are dark in color. We can see a darkly colored spherical dot inside the cell. This dot-like structure is called the nucleus. Let's try to see the nucleus of an animal cell. Using an ice cream spoon, gently scrape the inside surface of our cheek. You will now find a substance sticking onto the spoon. Now take a needle and a glass slide with water on it. Using needle, take the material that is stuck on the ice cream spoon and place it onto the glass slide. Then color it by adding a drop of methylene blue. Then add cover slip on top of it without forming air bubbles and observe under a microscope. We can see the cells. The cells will be like this. These are the cheek cells, and inside it, in the center, there will be a dot-like structure which is dark in color. This is the nucleus. The nucleus is covered with a double layered membrane which is called the nuclear membrane. Here you can see the double layered membrane, and this is called the nuclear membrane. And in the nuclear membrane there are pores. They are called nuclear pores. These pores will allow the transfer of materials to and from the nucleus. The center part is called the nucleolus, and the fluid inside this nucleus is called nucleoplasm. The nucleus has structures called chromosomes. This is a chromosome. This is a chromosome. We can see these chromosomes only when the cells are dividing. Cell division means one cell divides into two. This is a dividing cell, and here we can see the chromosomes. Here you can see that the cell is elongated, and now after this it will get split into two. And during this division, we can see the chromosome. These are the chromosomes inside the nucleus. When the cell is not dividing, the DNA present inside the nucleus is like a thread-like structure which is entangled with each other. When it is not dividing, the DNA is present like thread-like structures which are entangled with each other. This is called chromatin. This is chromatin. During division, we can see chromosomes, and when the cell is not dividing, we can see chromatin. But when the cell starts to divide, this chromatin material gets organized into chromosomes. Thus, we can see chromosomes during cell division. These chromosomes contain the information of characters or genetic material which are inherited from parents to next generation. For example, if your father has blue eyes, then there are chances that you may get blue eyes due to the transfer of these chromosomes which contain the genetic material. The chromosomes are composed of DNA and proteins. Let's learn the structure of DNA. DNA is the deoxyribonucleic acid. It is made up of two chains which coils around each other to form a double strand. This is one strand and this is another strand, and they coils around each other to form a double strand. The segments of DNA which are functional or active is called genes. In this DNA, this much portion is active and it can be called as a gene. These genes contain the information to specific traits, like as in our example, the blue eyes, and the transfer of chromosomes which contain this particular gene is responsible for the transfer of hereditary information. The nucleus plays a role in cellular reproduction. Reproduction means the cell organelles duplicate and two identical cells are formed. And it will determine the way the cell will develop and what shape it will obtain at maturity. Nucleus directs all the chemical activities inside the cell. But in some organisms, the nucleus doesn't have a nuclear membrane. This is a bacterial cell. Here, this is the genetic material, and these genetic materials are distributed in an irregularly shaped region. There is no nuclear membrane present here. This region is called nucleoid. This nucleoid is present in bacteria and they are known as prokaryotes. Prokaryotes lack a nuclear membrane. The organisms that have a nuclear membrane is called eukaryotes. This is the nucleus from a human cell, and here we can see the nuclear membrane. This is the nuclear membrane and they are called eukaryotes. Example of eukaryotes are fungi, plants and animals. Prokaryotic cell lacks most of the organelles which are present in eukaryotic cell. Many of the functions are performed by poorly organized parts in the cytoplasm. What is this cytoplasm? Let's learn about that. When we look onto the onion peel, we can see a large region inside the cell wall which takes a little stain. This much area is lightly shaded. This area is called the cytoplasm. We can see this cytoplasm in cheek cells also. Here, this whole blue area is the cytoplasm. Here the cytoplasm is enclosed by the plasma membrane. Inside the plasma membrane, cytoplasm is there. Cytoplasm is the fluid inside the plasma membrane. It contains the cell organelles. Each cell organelle has a specific function. In eukaryotes, that is the organisms with the nuclear membrane, the cell organelles are also enclosed in a membrane. Here these are the cell organelles and these cell organelles are also enclosed in a membrane. But in prokaryotes, which does not have a nuclear membrane, these membrane bound organelles are absent. Now let's look into the differences between prokaryotes and eukaryotes. Prokaryotic cell is very small, that is 1 to 10 μm in diameter, but eukaryotic cell is comparatively larger, 5 to 100 μm in diameter. A nuclear membrane is absent in prokaryotic cell, but nuclear membrane is present in eukaryotic cell. Prokaryotic cell has only a single chromosome, but eukaryotic cell has more than one chromosome. In prokaryotic cells, membrane-bound cell organelles are absent, but in eukaryotic cell, membrane-bound cell organelles are present. Let's learn about one of the cell organelle, endoplasmic reticulum. You can see this endoplasmic reticulum near to the nucleus. This is nucleus and this is the endoplasmic reticulum. It looks like long tubules. These are the long tubules. And the membrane of endoplasmic reticulum is similar to plasma membrane. There are two types of endoplasmic reticulum. Rough endoplasmic reticulum, that is RER, and smooth endoplasmic reticulum SER. RER looks rough under the microscope due to the presence of special kinds of structures on the surface. These are the special structures on the surface of rough endoplasmic reticulum. They are called ribosomes. Ribosomes are minute particles, consists of RNA and proteins. RNA is ribonucleic acid which is involved in the synthesis of proteins. Ribosomes contain this RNA and proteins. These ribosomes are present in all active cells and they are the site of protein synthesis. Proteins are essential for our body functions. These proteins are synthesized in ribosomes. As the ribosomes are present in RER, the main function of rough endoplasmic reticulum is the synthesis of proteins. The surface of smooth endoplasmic reticulum looks smooth as it does not contain the ribosomes. Smooth endoplasmic reticulum helps in the manufacture of fat molecules or lipids which are important for cell functions. Some of these proteins and lipids help in the development of cell membrane or plasma membrane. This process is called membrane biogenesis. What is membrane biogenesis? It is the development of cell membrane by the proteins and lipids synthesized by the smooth endoplasmic reticulum. So, as a result of membrane biogenesis, a cell membrane is formed and this cell membrane will help in the division of cell. Now, once again, let's look into the functions of RER and SER. Rough endoplasmic reticulum helps in the synthesis of proteins and transport of proteins to various parts of cytoplasm. Smooth endoplasmic reticulum manufacture fat and lipids for the membrane biogenesis. Detoxification of poisons and drugs are also a function of smooth endoplasmic reticulum. Moving on to Golgi apparatus. Golgi apparatus was first described by Camillo Golgi. It is also known as Golgi Complex or Golgi body. It is made up of membrane-bound vesicles arranged parallel to each other in sacs. These are the vesicles, and these vesicles are enclosed in a membrane, and they are arranged parallel to each other in stacks, and they are called cisterns. One is called cisternae and a group is called cisterns. We just learned that rough endoplasmic reticulum produce proteins. Golgi complex found near to endoplasmic reticulum. The proteins from endoplasmic reticulum buds off as vesicle from the endoplasmic reticulum's membrane. Some of these vesicles fuse together and form a large vesicle, and this large vesicle get attached to the Golgi complex. These proteins passes through the Golgi body and it get modified. Now the Golgi complex transport these proteins to different parts of the cells. Golgi apparatus thus package these proteins from endoplasmic reticulum into membrane-bound vesicles, and it transport these proteins to different parts inside and outside the cell. Now let's check what are the functions of Golgi bodies. It transports proteins to different parts of the cell. It helps to modify proteins. It is involved in the formation of plasma membrane and lysosomes, which we will study in later slides. It is an area of storage and packaging of cellular secretions. Lysosomes are membrane-bound sacs. They contain the digestive enzymes which are secreted by the rough endoplasmic reticulum. These are the digestive enzymes inside the lysosome. It is a kind of waste dispersing system. When foreign materials like bacteria and viruses enter the cell, they finally reach the lysosomes, and the digestive enzymes from this lysosome helps to destroy the bacteria and viruses. The old and worn out cell organelles also reach the lysosome. With the help of digestive enzymes, lysosome break the complex substances into simple substances. If the cell gets damaged, the lysosomes will burst and the enzymes inside it will digest its own cell. This is the reason the lysosomes are called the suicide bags. Why lysosomes are called suicide bags? If there is any damage happened to the cell, then lysosomes will burst and the enzymes inside the lysosomes will digest the whole cell. This is the reason the lysosomes are called suicide bags. Mitochondria is known as the powerhouse of the cell. It is called the powerhouse as it is responsible for extracting energy from food by converting sugar into energy. We know that the food we eat is split into tiny molecules by digestion. Mitochondria extracts energy from these sugar molecules which can be used by the body. The energy released is in the form of adenosine triphosphate ATP. ATP is known as the energy currency of the cell. Mitochondria extract energy from sugar molecules in the form of ATP. That is why ATP is called the energy currency of the cell. The body uses the energy stored in ATP for making new chemical compounds or for mechanical work. Mitochondria has two membrane coverings. As you can see, this is the outer membrane and this is the inner membrane. The outer membrane is porous and inner membrane is folded. This inner membrane consists of proteins which help in the synthesis of ATP. And the fold increases the surface area for the chemical reactions in the ATP synthesis. Mitochondria, unlike other cell organelles, has its own DNA and ribosomes. Because of the presence of these, that is the DNA and ribosomes, mitochondria can synthesize its own proteins. Plastids are present only in plant cells. In animal cells, we cannot see plastids. Two types of plastids are there, chromoplast and leucoplast. Chromoplast contain colored pigments. The chromoplast, which contains green colored pigment, is called the chloroplast. We know that the green colored pigment is the chlorophyll. In earlier classes, we studied that chloroplast is important for photosynthesis in plants. Chloroplast contains yellow and orange pigments in addition to this green pigment, that is the chlorophyll. Chloroplast contains numerous membrane layers. The colorless fluid inside the chloroplast is called the stroma. There is a fluid inside it that is called the stroma. And thylakoids, they are the membrane bound compartments. These are thylakoids. And these thylakoids form stacks of discs. It is called grana. Leucoplast is the storage area for starch, oils and protein granules. Like mitochondria, plastids also have their own DNA and ribosomes. Now let's learn about vacuoles. Vacuoles can be seen in animal as well as in plant cells. In plants, the vacuoles are very large and they contain full of cell sap. Cell sap is the fluid found in vacuoles. This will provide turgidity and rigidity to the cells. Turgidity means a state of being swollen. This is why the vacuoles are larger in plants. Vacuoles are of great helps to plants in deserts as it stores water. It also helps the plant to stand upright. In animal cells, the vacuoles are small. Many substances which are important for the survival of plants are stored in these vacuoles like amino acids, sugars, organic acids and some proteins. In amoeba, we earlier saw the food vacuole which contains the food that amoeba has consumed. With the help of all these cell organelles, the cell is able to perform cellular respiration, obtaining nutrition, cleaning waste materials and making new proteins. Due to this organization, cell is the structural and functional unit of a living organism. Now let's learn about cell division. Cells divide to form new cells. Due to this cell division, growth happens. The new cells which replace the old and injured cells. There are two types of cell division, mitosis and meiosis. Let's learn about it. Mitosis is a process by which the cells divide for growth. In this process, there is a mother cell and this mother cell divide to form two identical daughter cells. Here, this is the mother cell and this mother cell has 4 chromosomes and this cell divides to form two daughter cells and these daughter cells also have same number of chromosomes, that is in each daughter cell it has 4 chromosomes. Mitosis play an important role in the development of an embryo and it is important for the growth of our bodies. The old and damaged cells are replaced like this. Due to mitosis, growth happens. In meiosis, the cells of reproductive organs divide to form daughter cells called gametes. These gametes join to form the offspring. Here, this is the mother cell. This mother cell has 4 chromosomes and this mother cell divides by means of meiosis and 2 daughter cells are produced, but unlike in mitosis, this daughter cells has only half number of chromosomes. That means mother cell has 4 chromosomes, but daughter cells have 2 chromosomes. Half of 4 is 2. That's why the daughter cells, each of the daughter cells has 2 chromosomes, and these daughter cells again divide to form total 4 cells. But in this division, there will not be any change in the number of chromosomes. Each of this daughter cell has 2 chromosomes. This is the first division and this is the second division. In first division, the number of chromosomes are reduced. But in second division, the number of chromosomes are same. And these 4 daughter cells in the end, they are called the gametes. And these gametes from both individuals join to form a new offspring. The meiosis division is happening in the reproductive cell of humans. For example, in humans, a cell has 46 chromosomes, that means 23 pairs. This is the mother cell which has 46 chromosomes, and this mother cell divides to form 2 cells with half a number of chromosomes, that means 23 chromosomes. These daughter cells again divide and form 4 gametes with 23 chromosomes in each. These 4 cells are called the gametes, and the gamete or the reproductive cell in female is called the egg or ovum, which contains 23 chromosomes, and the reproductive cell or gamete from male is called the sperm, which has 23 chromosomes. During fertilization, they both fuse together. The egg and sperm fuse together. That means 23 plus 23, and the offspring which is produced as a result has 46 chromosomes. This is the reason we get the characters from both our parents, from our mother and father. That's it about the structural unit of life. If you like this video, please share it with your friends. If you have any queries, put it in the comment box. Thank you.